All right. Let's see. How many do we have here? 10 people, 11 people. Let's see. 17. Well, one thing that I wanted to tell you, and I would prefer that everybody's here when I announce this thing, because otherwise somebody's not going to hear it. I wanted to tell you that for the practical next week, I will give you a word bank for the regional terminology and anatomical terms. If you remember, in the back of chapter 1, there were certain sections that related to exercise 2 in the lab book. Remember, body organization and terminology. And there were all these terms that related to anterior regions and posterior regions. So I'll give you a word bank for those. Kind of like I did for the quiz, I think, right? Remember you had a choice of words to plug into those different figures? Do you remember that? Okay, so I will give you a word bank that relates to body regions. Okay. So I meant to tell you that before you left, and then I didn't think of it until after I got back to my office. Okay, so according to the schedule, you guys should be into or wrapping up the cellular metabolism chapter. Next week is when we start tissues. And we just had the test, obviously, today, so we can talk about whatever. We probably haven't gotten into the tissues chapter yet, so I don't know if there's questions about chapter 5. Probably not. We just wrapped up 4. So I'll just open up the room for questions, whether it's lecture or lab. What's on your mind? Since the lab practical is next week, we could talk about lab stuff. If you have any questions on lab.
So I apologize if you talked about this in lab this morning, but I was just, can you kind of go over what the lab practical will look like? Is that going to be like a mix of having to perform things on the microscope and paper? Will it all be a paper exam?
Well, we talked about that quite extensively this morning in lab.
Okay, that's okay.
And in essence, what I indicated was that when you walk into the room next Wednesday, we'll all start the practical at the same time. I'll hand out the question sheet and the answer sheet. And the majority of the questions will be non-station type of questions. For those questions that refer to stations, what I will do is I'll allow maybe 4 or 5 students at a time to go over into the bio lab where I will have the stations set up.
Okay.
And then you can answer those questions. I don't time you. You can go from one station to another station and back to the first station or whatever. The only policy I have is that no more than one person at any one time at any one station. So you may have to wait until a station frees up and you go check it out. The stations could involve microscope slides, microscope parts, any of the models we looked at. It could be a diagram, a figure in exercise 2 of the lab book on a body quadrant, similar to what you saw on the first lab quiz, I guess it was. And so you can look at the stations as many times as you wish. I don't time you, and that's kind of how we're going to do the practical. Most questions will be worth 1 or 2 points apiece. It's a 50-point practical, the first one. And I told everybody that don't forget Tuesday, the lab is open. So if you want to review models or slides, you can certainly come in and do that. So Paige, you weren't here today for lab, so you didn't look at the exercise 8 epithelial slides yet.
Yep, I'll be in next Tuesday to make up that.
Right.
Me too.
Yeah, right. So yeah, good that you asked that question, because you guys should know kind of what the policy or how it's going to kind of go next Wednesday, the format.
So this might sound like a stupid question. I don't know. So I understand that like the anaerobic, is in the like glycolysis, like part of the, process. Is that like the only, is the part where they're making like the lactic acid, is that considered part of the anaerobic, even though that's like down into the citric acid cycle?
Okay, let me pull up that PowerPoint.
I don't know if that's dumb or not.
No, it's not a bad question.
Just to know if I had to include that part if I was describing anaerobic, that part of it or whatever.
So this was chapter 4.
Yeah, like, I think 4.5.
Right. So let me let this get loaded.
That was pretty sweating.
You guys see the PowerPoint slides? Okay. So this was the slide. Where was it here? Glycolysis. This one, I think. Right? So what we remember about glycolysis is, again, that it takes place in the cytoplasm of cells in the presence or absence of oxygen. If there's no oxygen, if it's anaerobic, right? That's what anaerobic means, no oxygen. Then we would start with glucose, we would end with 2-pyruvic acid, and then that sort of defines the end of glycolysis, technically. So when we convert the 2-pyruvic acids into 2 lactic acids, that is sort of like a separate standalone set of reactions. But in essence, what's taking place is the NADH that was formed in glycolysis. Remember, this is a molecule that functions to transport electrons. It gets made here in glycolysis, but then its electrons are pulled off of the molecule and go into the production of the lactic acid from the pyruvic acid. So although you can't see that because we're looking at just the skeleton, if you will, of these molecules, the carbons, right? There are also hydrogens and other elements, if you will, connected to those carbons. So if we're talking anaerobic, the end process of respiration in cells is the production of this lactic acid. There's still 2 ATPs, a net gain of 2 ATPs. Okay, that doesn't change. You burn 2, you make 4, you come out ahead 2, right? But what is different is, of course, that we're talking about making a molecule here that can build up in muscles and cause cramps and that kind of.
Right. Okay. I guess I just didn't know if like there was any part further on that would be, I don't know, like that. So that's like basically the end of the anaerobic part is
Yeah, pretty much.
Lactic acid. Okay.
Right. If we go aerobic, of course, then we continue on to much more involves citric acid and electron transport. Right.
Like I understood the bus and the passer. Yeah. Okay. I just didn't know if like there was anything tagged that need to be tagged on elsewhere. I don't know.
Well, when we get into the muscular system chapter, which I think is like, I don't know, 9, we'll come back, we'll review this a little bit, and we'll talk about what lactic acid does to the body.
Okay.
So at least you have heard of it a little bit at this point, which is good.
Yeah, I just remember stuff from, yeah, okay. I just didn't know if I was remembering it right. Okay, thank
Yep.
you.
Yeah. Good question. Any other things like you want to talk about in chapter 4? And again, I know you've already had a test on it, so maybe there aren't any, but we can certainly talk about that. John, you're Mickey Mousing it again. It is just hilarious. I keep telling John he's got to pay his internet bill. Now he'll be fine, watch.
Am I still Mickey Mousing it?
No, you're fine.
All right, so I was saying anything that has to do with DNA replication and on, right? So genetic code, RNA molecules, protein synthesis, and the joining of the codons and what is anti-codon.
Well, this was part of chapter 4.
Yes, it is. It's towards the end.
Right, and didn't we put that on the test today?
Yep, I think so. I don't think we had anything.
I think the third Zoom video has it real good for chapter 4, cellular metabolism. It does break it down well. I watched it again right before the test. The third Zoom one I did.
Mm-hmm. So yeah, I mean the test you had, did describe some of that stuff. So you should be familiar with it. I mean, if you want to review any of it, we certainly can do that.
That
Can I ask
table.
one question? Oh, sorry. Go ahead.
The table.
Yeah, 4.2 table.
4.2 table. Let me look here. Oh yeah. Mhm.
Codons.
Right. So let me
Cells derive energy from the oxidate.
let me go back here and so the table that you're referring to is that table, right?
That table right there.
Yeah. What about it?
How does that?
How does that work?
Yeah.
Well, you have to understand that what this table is telling us is which of the 20 amino acids, and that's the name of these things here, these funny looking names, phenylalanine, leucine, isoleucine, methionine, valine, these are all amino acids, which you know are building blocks of proteins. So if a cell wants to make a protein, it needs building block molecules, which are linked together to make the protein. These 3 letters that you see here, these are called codons. And they represent the 3 nitrogenous bases within the messenger RNA strand that dictate which amino acid is brought in by the transfer RNA during translation. So let's take a look at that. On this slide, we see the messenger RNA here at the bottom. This is the messenger RNA strand. Now, what you are not seeing are the sugar phosphate groups that make up what's called the backbone of the molecule. That's not shown here. What are we seeing here? What are these things?
Your cursor isn't moving.
It isn't?
No.
Well, are you on this one?
Yep, there you go.
Oh, sorry. I got two PowerPoints, one on each. All right, sorry. So these things, what are these? Here's the sugar phosphate backbone. This structure right here, this line, if you will. And then what are these guys?
The RNA.
Well,
Right?
this whole thing is RNA.
Those are the nitrogenous bases that are the blocks of DNA. So then they match.
So these are the nitrogenous bases of the messenger RNA. Exactly right.
Okay.
And we see that every 3 of these is defined as a codon, right? Here's the first codon, AUG. Here's the second, GGC, and so on and so forth. And we have like what, 8, 9, if you count these guys, right? The reality is that it's not made up of 27 bases. Most messenger RNA would be hundreds and hundreds of bases long. Okay. So we're looking at just a little piece of the whole thing. What we're seeing here is the ribosome in brown. We have the big subunit here. We have the little subunit down below. This ribosome is made up of rRNA. What is that? Well, R stands for ribosomal RNA. So this is another type of RNA. It's called the ribosome. Okay, so just think of the ribosome is made up of ribosomal RNA. We'll depict it with a large and small subunit that has connected to the messenger RNA, because you can see they're connected. What are these funny looking blue guys? Well, follow the line, right? They're transfer RNA molecules. You got 2 of them here.
Uh-huh.
Now look at this one right here on the right, number 6. It's carrying the next amino acid that's going to be added to this growing polypeptide. Remember, a polypeptide is made up of many, many amino acids. Polypeptide is a protein, basically. So this amino acid, number 6, that's being transported by this particular transfer RNA molecule, how do we determine the identity of this amino acid? Which of the 20? How do we figure that out? Well, this is where that table comes in that you were referring us to a moment ago. How do we figure that out? Anybody?
By the codons.
Right. We have to go down to this codon sequence on the messenger RNA and see what it is.
See what the complementary one is, right? You match it.
Well, in order to tell us what this amino acid is, number 6, we have to look at the codon sequence of the messenger RNA.
Oh, okay.
Okay.
Now, and it is GCA, right? We'll go to the table in just a moment. But how did this transfer RNA get to this point? How did it know where to lock in place? How does that happen? Does any old one come in? No. What determines what this transfer RNA carrying this amino acid, what determines the specificity of this recognition system?
What it's matching into.
Right. It has to have the proper sequence of bases that complement the codon sequence. It's called the anti-codon sequence. So if we have GCA, obviously the anti-codon sequence must be CGU. Does everybody understand what I'm talking about?
Yeah.
If I'm talking French here, that's not good. You should be understanding what I just asked or what I said. If you don't, you need to be studying this more. So here's the next codon sequence, GGC. What will the anti-codon sequence be of the next transfer RNA bringing in the next amino acid?
CCG.
Say again?
Where CCG.
Yes, CCG is going to be the anticodon, right? How about the next one?
UUC.
TT.
No, 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 no,
UUC.
no T, no T, no T, no T, no T.
In RNA, it's uracil, so it'd be
Oh,
Right.
okay, all right, okay,
Don't forget, no thiamine in any RNA.
all right.
So in place of T, just put a what?
U,
A U. So what is it going to be? Anticodon?
U,
UU what?
C.
Right. Right. How about the next one? Anticodon.
GCA.
Right. Everybody see that?
Mhm.
Okay, so back to this transfer RNA. Here's the codon sequence. Here's the anticodon sequence. I want to know what this amino acid number 6 is. In order to determine that, I got to look at the codon of the messenger RNA, CGA, 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 CGA.
Arginine. Is it arginine?
Where is it?
Arginine.
CGA. There it is. Yep. Arginine. That's number 6 amino acid, arginine. Right. How about the amino acid here of 5? Let's look at what's this guy? ACG. 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 There it is. Threonine. So the sequence of amino acids, which determines the shape and function of the protein, is determined by the codon sequence of the messenger RNA, which was ultimately determined by what?
The DNA.
Right, the DNA. DNA was the ultimate determinator of what kind of protein was going to be made by that cell. Yep. Now, you also notice that on that chart or table, there were 3 codon sequences that did not specify for a given amino acid. These are the 3 stop codons. What is their significance in the translation process? How will it Go ahead, Sadie.
That's when they like stop bringing in more transfer RNA. It like cuts the amino acids. It's like the last one.
Yeah, it basically tells the system, if you will, we're done assembling the protein. No more amino acids will be brought in because there's no anticodon that's going to recognize that. Stop codon. You know, it stops translation. We're done making the protein. Now, what if there's a mutation such that a UAC is altered and we change this last base from a C to an A? Ordinarily, tyrosine would be brought in, but now because of the mutation defined as a change in the DNA, which results ultimately in a change in this base of the messenger RNA, what's that going to do to the protein being produced?
It would stop it too early, maybe.
Yeah. If it occurs early in the sequence, you're going to be making a malformed protein. It's not going to be functional. Yeah, absolutely. Now, it's interesting to note that some bases, let's just pick this one here, CCU. Let's say we change this U, mutation comes in, pulls that guy out, we put a C in there in place of it. What's that going to do?
Are you talking about like the disease it'll give you?
No, I'm asking what effect will that have on the protein being
It's
made?
gonna make it malform. I mean,
No, 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 no.
It's
If
just-
I change the U here, do you see that on my pointer?
Yeah.
I change that U to a C.
It'll still bring the same thing because they're in the same column.
Right. That's not going to have any impact because it's going to bring in the same amino acid. So do all mutations necessarily bring about a problem in the cell? Potentially. No, not always. Not always.
I mean, mutations like really the basis of evolution, right?
Exactly right, Freya. Excellent point. That's very intuitive. Now, a lot of people don't understand what you just said because they haven't studied this, but you're absolutely right. Mutations are the source of microevolutionary processes. Absolutely. And it's quite interesting if you study it enough and think about it. It's fascinating. This
It could bring,
this
is
is at
that,
the cellular level, okay?
that brings about diseases? Well, that,
It could, sure. There are a number of diseases, inherited diseases whereby, say, sickle cell anemia or sickle cell disease, right?
Okay.
It's very, very common in the African American community, more so than in Caucasians, right?
Right.
And I talk about that in the end, toward the end of the chapter, right, where we discuss how changing just one base here, okay, the lightning bolt obviously is sort of a symbol to try to indicate, well, there's a change in the DNA. And we change that TAC to, let's see, I'm sorry, we changed the TTC, we changed this middle T to an A, okay. We remove the T, we put an A in, the mutation does. And you're saying to yourself, well, okay, we're still bringing in an amino acid. It's not like we're introducing a stop codon or something, right? No, it's not the same amino acid, but hey, will anything else be affected downstream from that mutation?
sure.
Answer is no. All the same amino acids will be brought in. So we're talking about this huge, huge protein, okay, of which only one amino acid is different. How can one little amino acid make a difference? Well, it can. It can make the difference between having normal shaped red blood cells versus sickle shaped red blood cells. Yeah. What we have going for us is the fact that when DNA is replicated, which is a different process, but you know what I'm talking about, right? During interphase, S phase of interphase, the DNA unzips and then the new DNA bases come up and we make the new 2 double helices, which are divvied to the 2 daughter cells. When that process occurs, our cells have special proofreading enzymes that can scour the newly replicated DNA. And if they encounter a mismatch, like a C opposite a T or an A opposite a G, for example, which would be not correct, the newly synthesized base will be removed. We remove the G and we put a T in because that's what should be opposite A. So that's what the last part of the last, I think, slide talks about, these amazing proofreading enzymes that are there to catch those very rare mismatches, which could be problematic down the road. But as we know, as one gets older, our cells are not as efficient in catching every single mutation, and they escape detection, and things can happen, and things like cancer can occur and other problems.
And like it's more like in scar tissue and stuff, and more likely to like mutations and stuff.
Are you asking?
I mean, would it be more likely to have like, you know, cancer like to develop from like scar tissue and things like that, I guess? Like
No, I don't think so.
if scar tissue doesn't mutate more likely. Okay.
No, I don't think so. Not that I've ever heard. Yeah. Good questions. I don't know, John, you brought us to that table. Were there other questions about it?
Now I'm gonna give somebody else a chance.
Any topics in chapter 4 you'd like to talk about? Or we're kind of in that quasi middle thing zone between ending material and starting new stuff. We could talk about the chemistry exam too. If you had any questions on that, we could talk about that if you want. Since not everybody may have had a chance to look at the answer key, we've got time now if you want to go over that. I'm just throwing it out as an idea. If you don't, that's fine too.
I quickly read through chapter 5 and I had some questions on that, but I don't want to jump ahead if nobody else has read that.
Well, we started into epithelial tissue in lab today, so but if there's no questions on 4 or whatever, certainly we can talk about 5. So I'd say go ahead and ask, Paige.
I just had a quick question about the like intercellular junctions and like how the epithelial kind of like holds itself together. Like the like I don't know, like is it the actual like cells linking together, or it's like a substance holding them together?
Yeah. Okay, so let me just say from the outset that, and I'm going to pull the PowerPoint up here. Do you see that now, the PowerPoint?
Mm-hmm.
Okay, so I don't specifically talk much about intracellular junctions and don't get into the details between knowing the difference of a tight junction versus a desmosome. So I am not going to ask you anything on intracellular junctions.
Okay.
Just kind of FYI. So in the future, which I applaud you for looking ahead, that's very admirable. I appreciate you doing that. If you do that in the future, allow the PowerPoints to sort of guide you in terms of the material.
Okay.
But I would always ask, right? And if not sure, ask. But there are occasions when I don't always include everything that the chapter presents. And case in point would be the very first couple paragraphs of this chapter, which you caught. And then also, as long as we're talking about it, in the epithelial section, toward the very end of it, on page 157, there's some discussion of glandular epithelium. Okay, and this is the slide that I put on the PowerPoint. I do not get into discussing merocrine, apocrine, holocrine glands, nor do I get into the various kinds of structural examples of exocrine glands, like on page 158, figure 5.12. If you happen to have your book, you can flip to that, page 158, the bottom half. So that's not something you need to worry about for lecture. And the same applies on page 159, that table 5.3 or the figure 15.13. Just kind of FYI, and I'm glad you brought up that question. I'm pretty sure in the Zoom lecture I probably say that, but as you begin to look at chapter 5, and you certainly should, those are a couple sections that that I did not include, and you don't need to worry about. But I think certainly for the practical next week, you should be getting into chapter 5. It does a nice job reviewing the epithelial tissue like we just talked about not more than 2 hours ago. So this is going to be fresh in your mind. You guys that came to lab, you're going to be like, oh yeah, I remember that. Now, Paige and Beth, it's going to be new for you, but by looking ahead and being familiar with this, when you get to lab on Tuesday and look at those slides, hopefully it's not going to be nearly as bad as if you hadn't looked at it. The other thing that I told everybody on today in lab was after the practical next week, we're going to wrap up the connective tissue, the muscle and nervous tissue slides. And so it would really be great if you could try to get through this chapter by Wednesday. Now, and I know you're focusing on the practical, so I get the fact that you only have a certain number of minutes in a day and all that. But if you can look ahead, it will make for less confusion in exercises 9, 10, and 11 that we're doing next week. I mean, we looked at what, 6 slides this morning, I think it was, something like that. And I think we spent at least an hour and 15 minutes or more on just 6 slides. If you remember it, we spent a lot of time. So next week, when we do 7, 8, and 9, right? And I don't have the handout right with me, but there's like 3 times as many slides, approximately, maybe. So we're going to be moving through those slides. I won't say like really rapidly, but we're not going to have 2 hours to do the slides. We're going to have to get through them, and we will. But by looking ahead and becoming a little more familiar, we'll make it much more understandable, I think, for you next Wednesday. How about other questions? No questions?
Nothing on tissues is going to be on the practical, right?
Wrong. We said that the stuff we did today was going to be in the practical next week.
Okay, I was just double checking.
Because we included exercise 5, I think it was. I'm sorry, not 5, 8. 8 is the first tissue exercise. So it's exercises 2, 4, 5, 6, 7, 8. And the rat dissection. Don't forget the rat dissection.
Okay, I just had a brain fart. I'm just making sure.
Yeah, do not forget about the epithelial tissues. There will be some questions on that. And again, you can always go to this syllabus, right? It's all there in terms of what exercises are going to be covered. Now, I did erroneously put exercise 1 on here, which John caught this morning. So you can cross that 1 off there, right? That was a type 1 on my part. No questions, no comments?
Hmm.
Don't be afraid to ask.
So what we gonna do for the quiz on the rat dissection? You will ask about the organ system of the rat.
Well, the rat dissection will be on the practical next week. So you need to go back and review the diagrams from the handout, right? And then there's also that preserved rat in that plexiglass model in the front of lab. Do you remember there was the square box that had the fluid inside and the rat was in the fluid? I showed you that the day we did the dissection. I held it up and I said, you know, you can always look at this rat and it's got the organs numbered and you can look at the key. There's a little key, answer key on the back of the model. And there's also, I think, a little laminated answer key that Deb had put on the top of the model, but you'll be able to see the key there somewhere. So you could see the rat as a station. I could get one of those rats out like I dissected for you and open it up and pin it down and put some needles numbered, asking you to identify an organ or anything we may have talked about. So yes, the rat is fair game for the practical. And I know you have just the diagrams to look at, but you do have the specimen as well in lab in that box. Does that answer your question, Maiving?
Yeah, this day. Thank you.
Sure. Other questions?
So exercises 2 through 8 in the lab book are going to show up. Okay.
Right. I've got the Zoom till 2:30. I'm not sure how it's going to go. Are you around or? Okay. I can't do anything right now, but stop by in 15, 20 minutes. Okay. Yeah, so what we've said, you know.
Oh, got it.
There's not an exercise 3, so it's 2, 4 through 8 in the rat dissection. Okay, well, I will be here for the next half hour if you want to stay in the room and ask any questions you've got or whatever. Don't forget the lab's open Tuesday if you want to come in. Kyra's doing her Sunday evening review still. I'm not sure how many of you have gone to that. I know, John, you've gone to one of them, or maybe more. How many have gone to Kyra's thing?
I do.
Okay, and so can you tell the group, how she does that? Has it been helpful?
Sure. Let me start my video. So I go every Sunday. It's from 8 and yes, it's been helpful. She kind of, right now we're doing like a little game I think she's starting to do where you have to download the app on your cell phone. And then you just kind of, she asks questions and then you answer it. So she makes it kind of so it's not boring or just, so it is fun and it does help. It is a fun study group. Like I do get a lot out of it, especially last class we went over like all the cells and and it helped a lot. So I definitely recommend it if you haven't tried it.
And how many people are showing up to her reviews approximately?
I think I've seen 3 from our class.
Okay.
Probably more. But I think there's at least 14 to 16
I don't know if there were that many.
altogether.
Was there?
No, not that many. I thought last class there was.
I'm not sure, but the games were fun. I thought they were kind of cool. It was like, what was it? Kaboot?
Yeah.
I don't know. And it was like
Yeah,
asking questions.
yeah.
It was neat. And then
Yep.
there's like, you can do your own flashcards on these different websites. So there's a lot of other tools that are on these websites that you can use at other times that are awesome.
Yep.
Yeah, what was it, honey? Quiznet?
Quizlet.
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 And the other one was like, started with a K. It was pretty cool.
I usually get her one-on-one tutoring. So I go to Starfish and then I sign
Make an
in.
appointment.
So I see her a lot that way, and she is super smart.
Yeah, she's a good student.
Is that the review that you have to sign up for?
Are you talking about Kyra's? Yeah, I think she just allows you to pop in on, it's 8 p.m. on Sundays, right? And I send you guys the link, I don't know, 2 or 3 weeks ago, but so you can go back and get that. Does anybody have her email or Blackboard, or not Blackboard, but a Zoom link?
I have the email.
You do?
Go back.
Okay, Brittany, so just if you can find that email that has the Zoom link, just come, a couple minutes before 8, and
You just had to put that password in or whatever. It's like capital A and P group. It'll ask you for.
but that passcode was part of what she had sent.
Yeah, it came with that email. Yep.
Yeah. Okay, good.
Just why I'm kind of slow at this, so it took me a minute.
Well, unlike mine, where you just kind of click and you're there, I just tried to make it as simple for you as I could. I didn't want to include a passcode and all that stuff, but just an added security thing, right? So you won't get anybody. What's the term? Zoom bombing you or something? Right? Is that the right term? Or somebody pops into your room?
Yeah.
Just want to jump into physiology and anatomy for the afternoon.
Thank you, Jolene, for that update. That was great. So yeah, I'm certainly getting a sense of people who have gone have enjoyed the experience and have benefited from it. And as you said, she's just very up on things. She's a natural tutor. Yeah, she's a good student. Speaking of good students, one thing that we're trying to do in the science department, on both campuses, is advertising these career communities. You may have heard of career communities. We're trying to get students who come to JCC to give us a sense of kind of what they want to do down the road in terms of a general career, and then we try to plug them into these career communities where you get people of similar interests getting together and talking about, fields. And one of the science faculty is kind of heading this up. So every week or two, he's highlighting a former alumnus who's doing something in the science field, just to give you guys a sense of well, this is where what you can do with your life too. Here's what so-and-so has done, right? So I was kind of brainstorming a little bit. Who have I had as a student years ago that has really, gone places? And I came up with a student who I had probably, oh gosh, 12, 14 years ago. His name is Colin Kramer. He was homeschooled, and both his parents had gone to Cornell for their undergraduate degrees, and they homeschooled he and his sister from pre-K up through high school, I think. He came to JCC, and he just excelled through his 2 years here. He went to Geneseo, got his bachelor's degree, and then he went to Yale and got his PhD, and then he got his postdoctoral work at like Harvard or Stanford or somewhere, and now he's a professor at UCLA.
I had him for a tutor.
You had him for a tutor?
Yeah, in the lab there.
Where? At Geneseo?
Nope, here, JCC.
Oh.
Yeah, we were doing business, like accounting.
Well, I don't. He was in biology. I don't.
Yeah, but he knew a lot of stuff.
When? A long time ago, you mean?
Yeah, this was a while ago.
Well, Colin's been out of. I don't know when he graduated from JCC.
Yeah, this would have been 2006.
Oh no, it's not the same person.
Okay.
No.
You must have had a ton of students though over the years, right?
Yeah. Oh yeah.
You said 35 years, I thought you said.
Yeah,
That's
yeah.
crazy.
It's hard to believe.
You probably know every trick. I taught that's my degree is English teaching.
Yeah.
If I talk too much, it's because I feel bad when no one answers. So I just like, I wait and I'm just like, because I taught before. So then I just feel like you're like hanging. I feel bad. Sorry if I do.
Well, again, the point being that, JCC can act as a stepping stone for lots of
That's
people.
crazy.
And, he's just an amazing person. And he's just been there a year. So he's green. He's very green. But I mean, what a amazing career he has ahead of him. I mean, UCLA is like one of the best schools in the country. And he's probably by now in his early 30s, but to get to that place is amazing. So, that's pretty cool. So I want to be able to say, oh yeah, I had, Paige or Sadie or Nikki or Jolene or whoever, in 5 years, and now they're doing this. And yeah, I mean, you all will have amazing step ladders to different disciplines. It's always fun to see people go places.
Will he be able to find anybody that's in my field?
You know what?
Will he be able to find anybody that's going into my field? Because it's almost like unheard of.
I don't know. Coding?
Yeah.
That's sort of science and it's kind of business, or I don't know. I don't describe coding. Yeah, so.
It's both. You can't do medical coding without A&P.
Sure.
Might as well not even show up for work. Forget about it.
Yeah, you got to know the language, the lingo, right?
You got to know what the doctor's talking about and what they ordering and what they've done.
Yeah, right. Any more questions before I let you go? Okay, great. Well, I will see you next Wednesday. Bye.
All right, I got a question for you.
Okay.
All right, so it's still on the practical, okay?
Yeah.
I hope it don't sound redundant, but the cells, chapter 2 quiz. That's going to be on there. And how much is that? Is it going to be the same stuff?
Okay, when you say chapter 2 cells, tell me what that means. Are you talking about exercise 2 in the lab book? Or chapter
Chapter 2 in the book.
chapter 2 in the textbook? That's the
So if we do chapter 2 in the lab book, right?
okay, when we say chapter, we're talking textbook, right? So exercise would be a lab book.
Yeah.
So if you're talking about the cells, okay, which you were in chapter
In chapter 2,
okay.
right? That's going to show up.
Well, chapter 2 is chemistry. Chapter 3 is cells.
Okay, that's right. Okay.
So you want to ask about the cells chapter?
Yep, cells chapter is going to show up on the practical.
The chapter 3 cells stuff that we covered in lecture is lecture stuff. The only thing we talked about in the lab related to cells was exercise 5.
5, okay.
So let's take a look at exercise 5. Cell structure. What's the title of it here? I forget. Cell structure and function, I think.
I'm almost there. Yep, cell structure and function.
Okay. So as you look at that exercise, it starts with cell structure and function.
Right.
There's a diagram of the cell, right? This is the same diagram that you have in the textbook in chapter 3. So you should, for the practical next Wednesday, be familiar with the organelles and what they do. You should have already learned that because we just had the test on it today. So that is going to be review. You should be able to recognize what the function of the mitochondrion is, or what the Golgi body does, or what cilia do, where they're located. So by reviewing the lecture, chapter 3 lecture stuff on the cell, you are preparing yourself well for the first part of exercise 5.
All right. Chapter 3 out of the book. Okay.
Right. So there's overlap between lecture and lab in that case.
Hang on. Excuse me. There it is. I got it. Then chapter 4. It's not 4. Lab exercise 4. Let's use a microscope. Okay, good. I got it. I'm sure I'll be hitting you up again.
So back to exercise 5 for just a minute. In addition to the cell diagram, the book also reviews the cell membrane. That's what this is.
Mm-hmm.
You've already learned about the phospholipid bilayer and embedded proteins and what they do. If you think back to the Zoom lecture that I did for chapter 3 in lecture. So you've already studied that. So there's really nothing new.
It's just reviewing it.
Yeah. Now, what we also did in exercise 5, I'm sorry, 6. 6 was movements through membranes, which gets at what function membranes have, and that is to regulate what enters and leaves through the cell membrane. So we did this last week. This is where we looked at diffusion of the potassium permanganate through water. Remember, we put it in the fecal
Yep.
plate?
Mm-hmm.
This is where we looked at the movement of water through that semi-permeable membrane. That demonstration I set up with the dialysis tubing and the 1 ml pipette in the beaker of water, right? So you should go back and review what we did in exercise 6. I also had Freya come up and she donated some drops of blood. And
Yeah.
we subjected that blood to 3 different salt solutions. And we looked at what effect those salt solutions had on the integrity of the red blood cell as it related to water movement in or out. That's definitely going to be on the practical next week. So you should go back and review hypertonic, isotonic, hypotonic solutions. What are those solutions? What do those terms mean? Where does water move? Well, you've got to ask yourself, where is water more highly concentrated, on which side of the membrane? Because water will always move toward a higher solute or toward a lower water concentration. So this is stuff we talked about last Wednesday. That's all stuff you got to spend some time reviewing and learning, because that'll be on the practical.
Glad we're recording this. And I'm familiar with 7 because we just went through that. And then 8 we're on now. Okay.
8 we did today. Yep.
Yep. So I'll put on some jazz music and I'll just get busy.
Okay, sounds good.
Hey, you have a good one.
Okay, John, you too.
Hey, thanks.
You bet. Bye.
Bye.
Hi, Jay.
Hi.
Hi.
I have the question for the lab quiz number 2,
Okay.
the number 8.
What was the question?
Function to carry from each kidney to the blank.
Oh,
The function.
that was question.
Is it the question on the rat diagram?
Hang on. Was this question number 8, you said?
Yeah, number 8.
Yeah, 8. Okay. So the blank function to carry blank from each kidney to the blank, right?
Yeah.
And what did you put in there?
I don't know the function, but I say it carry the fluid from kidney to the urinary.
Can you hold your quiz up to the camera? I can't.
Yeah.
I can't understand. Oh, fluid to the urinary. Okay, gotcha. All right. Hold it one more time, please. Hold it up there one more time so I can see it. Can you hold it up? Take your quiz and show me again. Yeah, let me see. Okay, I took one off. Okay. All right. So The answer to that question, did you write down the answer?
I
No.
didn't hear that.
Oh, okay.
So,
So the answer to the first blank is ureters. Ureters.
urethra.
Here. Ureters. Or ureters, if you either it's pronunciation.
Ureters.
Those are the tubes that come from each kidney toward the bladder.
Bladder.
So the ureters carry urine. Now you said fluid, right?
Yep.
I will give you half a point for fluid. But how many different kinds of fluids do you know? Water,
Water.
coffee, Coca-Cola,
Yeah.
blood. There's a lot of different kinds of fluids. So when you get a question like this, and it says from each kidney, right? From each kidney, coming from each kidney, you're not going to put Coca-Cola, right?
Yeah.
So you want to be as specific as you can. You said fluid, which is true, but do you think urine is a better answer?
Yep.
Yeah. So I would say try to be as specific as you can. Rather than say fluid, which could be thousands of different things, we know that if it's coming from the kidney, it has to be urine.
Urine.
Urine. Does that make sense?
Yeah. And to the bladder.
Bladder.
Okay.
Urinary bladder.
Bladder.
Here. Urinary, which is the name of the system, right? Urinary system. Bladder is the bag, if you will, that the urine will collect in.
Yeah.
So is it the English that's causing the problem? The terminology, the English, is that difficult? You didn't know?
Yeah, I didn't know the meaning.
The word.
Yeah.
Yeah. Do you know the word in your native language? In Vietnamese, is there a name for the tubes that come from the kidneys? There has to be some term.
Yeah.
If you're a doctor, you have to tell your colleagues, your patients, the nurses, what is that tube called?
Yeah.
No name you can think of, huh?
No. Actually, I don't know this question. And for the question number 4,
yeah.
Can you explain it one more time, like
Sure.
as the working distance
Yeah.
decrease and
Yeah. So it says as the working distance of the microscope decreases, that means getting smaller, right? Decrease meaning getting smaller, increase getting bigger.
Yep.
Yeah, we're good with that?
Yeah.
Okay. Working distance, we defined that when we looked at the microscope 2 weeks ago. And we said that the working distance is the distance between the objective and the slide. Do you remember us talking about that?
Yeah, I remember that.
So let me find it here. So there it talks about the working distance.
Distance. Yeah.
Okay. So as that gets bigger, I'm sorry, we're number 4. As the working distance gets smaller, what happens to the magnification?
Oh, it will bigger, increases.
Right,
In the objective.
right.
Yeah, I confuse between the number 3 and number 4.
Oh,
So, but I got the number 3.
you got number 3 correct?
Yeah, yeah.
Was that a lucky guess?
Huh?
Was that a lucky guess? Did you just say,
I,
oh,
no,
did you
I
know
got,
it? Did you know it? Or did you not sure? You just circled.
no, I get it wrong too. I got it wrong to the answer too.
You got 3 wrong.
I got 4.
Number 3 was correct.
No, it's wrong too.
Okay, so it says as the magnification increases, the field of view, what you see when you look through the scope, the field of view, what happens to that? As you go under higher magnification.
Before I thought like the field of view. This means you can see the what's it that called? The one in the slide. You can see it closer. So I circle the increases.
No, if you are magnifying something, you're zooming in on a smaller area. So the field of view gets smaller. What you see inside that is smaller. It's magnifying it. So you're, it's like saying, how do I describe this?
So you mean the field of view, it means the space.
Yeah.
When you see in the microscope, it will smaller, right?
Yeah. What is inside that space in that field of view is smaller because you're zooming in. You're magnifying in.
Yeah.
Okay?
I have the different thinking like
Yeah.
because I think the field of view it means when you zoom the microscope like closer, you can see the sample more clearly. So that's why I circle increases. You understand what I mean?
Oh yeah.
Yeah.
No, I understand what you were thinking.
Yeah.
But the field of view is the whole thing. It's not just one item.
Okay.
This is a language thing. I think you're having difficulty with. So if you look at, let's say this is a field of view. And let's say we're looking at bacteria. So let's just pretend we're looking at some cells, okay? And there's like 2, 4, there's 8 cells there, right? 8 bacteria. And let's say that this is low scanning objective. So 40 power, 40x, 4 times 10, 40. Okay, now we go to low power, which is going to be 100, 10 times 10, right? We're only going to see 2 cells. Now this circle here that I drew, the little one, that still looks the same size when you look through the scope. But what is inside of it has less cells because you're zooming in.
Yeah.
If you go to high power,
The
maybe
area
you
smaller.
only see one cell because you're zooming in. When you look through the scope, the size of the field of view, you know, the circle is the same, but what is inside of it changes because you're zooming in, zooming in, zooming in.
Yeah.
Does that help? I don't know if that.
Yeah, I understand it now. Thank you. And for the number 6, where is mesentery? Is it the
Mesentery.
orange or is it something?
It is a membranous material. It looks like a membrane.
Membrane.
Yeah, let me show you. Hang on one second. This is the mesentery. This is the small intestine. This membranous material with blood vessels and some fat. This is mesentery.
Where is it in the body system? Is it?
This is the small intestine. The
Oh,
mesentery
small
helps to hold your small intestine together. It's very highly coiled. And there are blood vessels that absorb nutrients from the small intestine and take that into the general circulation.
Oh, okay.
So that's what question 6
Yeah.
was asking for.
So for the chemical to the exact the chemistry exam, how you count, how you will grade it into the total final score?
Oh, when I go to calculate the final grade for this course, I add up all of your points from lecture and lab. I add them all together, and then I divide by the total possible. So if you want to know where you are right now, you got a 9 on the first lecture quiz, right?
Yeah.
23 on the chemistry exam, 8 on the first lab quiz, and 6 on the second lab quiz.
You divide it to 4.
So we take 9 plus 23 plus 8 plus 6 equals 46. Is that right?
Yep.
46. And that is divided by 60.
60.
60 possible points.
Oh, yeah.
10, 30, 10, 10.
So you use 46 divided by 60.
Yeah. And that equals a 77%.
Yeah.
So I'm just adding, I'm adding up your points and I'm dividing by the possible points. So there's a possible 60 points you could have gotten on the first lecture quiz, on the chemistry exam, and the first 2 lab quizzes. You got 46 out of 60.
Yeah.
We're at 77%. So that's where you are right now.
Mm.
I have not corrected the exam you took today, so we would add that to the 46, depending upon what score you got. But then 60 becomes 140 because we have the 80-point exam.
Yeah.
Does that make sense?
Yeah.
Okay. So if you want to see what your grade is, all you have to do is add up all of your points and divide that by the total possible. And then you can go and find the percent. And then you can go to this table.
Yeah. Okay. I think I didn't do well on the this morning exam.
You don't?
Yeah, I don't.
Well, we'll see. Maybe you did better than you think.
Oh, I don't know. Maybe half of them almost. I'm not sure about the answer. Is it possible like if for the second one, no, for the second exam and the next quizzes, if I do it better, I can get an A?
I think we should see how you did on this first test. Then I can tell you an answer.
Yeah.
I can't say until I take a look at what your score was.
Yeah.
But next week we can talk.
Yeah.
Okay.
Okay.
And the other thing that I was going to suggest too is if you would like to talk to Don Poole, did we talk about Don?
Yeah.
Don Poole?
Mhm.
If the English is causing problems in terms of you understanding what's being asked, then maybe we need to bring Don Poole into our conversation and see if he can assist us, assist you. So let's see how you did, and then we can talk about that.
Yeah. Okay.
Okay?
Yeah. Okay.
Okay, sounds good.
Thank you.
You're welcome. See you later.
Yeah. Thank you so much. Have a
Sure.
good one.
Thanks, you too. Bye.